In this video, we will discuss 8 hallmarks of cancer. So, let's get started. 6 acquired functional capabilities of malignant tumors were defined by Hanahan and Weinberg in 2000 and in 2011. The same authors proposed 2 more, plus 2 enabling characteristics. Let's discuss these 8 hallmark abnormalities, common to the 100 or more kinds of malignant cancer. First is self-sufficiency in mitotic stimulation. Second is evasion of growth suppressors. Third is resistance to apoptosis. Fourth is limitless replicative potential. Fifth is sustained angiogenesis. Sixth hallmark is tissue invasion and metastasis. Seventh is reprogramming of cellular energy metabolism. And the 8th hallmark of cancer is evasion of immune destruction. Now let's discuss them one by one. First hallmark of cancer is the self-sufficiency in mitotic stimulation. Let's discuss it in detail. In mitosis, growth factors are normally required for mitosis, and these engage with growth-specific receptors on cell surfaces. Cancer cells, however, can grow and divide without external signals. So cancer cells has ability of utilizing 3 unusual capacities. Now let's discuss these 3 unusual capacities of cancer cells. First is the production of self-stimulatory growth factors. Second unusual capacities of cancer cells is hyperexpression of growth factor receptor. And third is the spontaneous transmission of intracellular mitotic signals by unstimulated growth factors receptors. Moving to the second hallmark of cancer, and that is evasion of growth suppressors. Normally, in the G0 and early G1 phases, retinoblastoma protein is a tumor suppressor gene and critical cell cycle regulator that targets the E2 promoter binding factor 1 factors family of transcription factors. Retinoblastoma protein physically associates with E2 promoter binding factor 1 factors and blocks their transactivation domain. In the late G1 phase, phosphorylated retinoblastoma protein releases E2 promoter binding factor 1. E2 promoter binding factor 1 is the founding member of an evolutionarily conserved family of transcription factors that play critical roles in the regulation of the cell cycle and apoptosis, allowing the expression of genes that encode products necessary for S-phase progression. Cancer cells generally resist growth inhibitory signals from the extracellular matrix and neighboring cell surfaces. Normally, such signals are transmitted internally, ultimately through the retinoblastoma protein and others, preventing inappropriate transition from G1 to S. But, in case of cancer cells, this transition is blocked by underphosphorylated retinoblastoma protein, which sequesters the necessary E2 promoter binding factor 1 transcription factors. But, do you know such factors that cause this? Let's take an example. Transforming growth factor beta inhibits the phosphorylation that would inactivate retinoblastoma protein. If the action of retinoblastoma protein is harmed by mutation or upset by intervention of papillomavirus in uterine cervical cells, uncontrolled cell division can lead to cancer. Moving to third hallmark of cancer, and that's resistance to apoptosis. Let's discuss it in detail. Accumulation of tumor cells depends also on their rate of attrition. In normal cells, sensor molecules check for metabolic abnormalities before effectors activate the death pathway or apoptosis. The sensors include insulin-like growth factor 1 and insulin-like growth factor 2 and insulin-like growth factor 1 receptor. But, do you know the function of these sensors? Well, they monitor DNA damage, proto-oncogene expression levels, hypoxia, and signals from the extracellular matrix and cell neighbors. Now, let's discuss the effectors. The effectors include FOS ligand and tumor necrosis factor alpha and their receptors, which are FOS receptor and tumor necrosis factor alpha receptor. Apoptosis is normally triggered by overexpression of a proto-oncogen, and a cancer can progress to malignancy only if that is overcome. Moving to the fourth hallmark of cancer, and that is limitless replicative potential. Let's discuss it in detail. When multiplication is carried out with human cells, the Hayflick limit restricts their multiplication to 80 times. The limit is normally imposed by proteins retinoblastoma protein and p53. But in cancer, retinoblastoma protein and p53 are disabled. So, cancer cells ignore this restriction of Hayflick limit of multiplication to 80 times. So, as in typical cancer cells, mitosis continues until a stage called crisis is reached, resulting usually in death of the cell. Moving to the fifth hallmark of cancer, and that is sustained angiogenesis. Let's discuss it in detail. Tissues need capillaries within a range of 100 micrometers, and initiation of growth of capillaries called as angiogenesis. This angiogenesis requires both inducers and inhibitors. Inducers include vascular endothelial growth factor and acetic and basic fibroblast growth factor, which bind to tyrosine kinase receptors on endothelial cell surfaces. But what's the role of cancer in this? Well, in case of cancer, vascular endothelial growth factor expression is overactivated by the RAS oncogene. RAS oncogene are mutated and changed form of a type of gene called as proto-oncogene. Now let's discuss how inhibitors are affected in cancer. Well, thrombospondin 1 is an angiogenesis inhibitor, regulated by p53. But in cancer, p53 is disabled. So, does the regulation. All these defected factors ultimately affects the angiogenesis. Moving to the sixth hallmark of cancer, and that is tissue invasion and metastasis. Let's discuss it in detail. Tumors are of two types: benign and malignant tumor. However, most tumors are benign tumor. These rarely present a threat to their hosts, it do not invade and not metastasized. Whereas, malignant tumors has ability to invade other tissues and to metastasize. When cancer causes death, this usually results from invasion of neighboring tissues, frequently following dispersal to distant sites. Tumors of both types are found in the same organs, can be derived from the same cell types, can grow to the same size, be induced by the same agents or inherited mutations, or arise spontaneously. The major distinction of malignant tumors is their ability to invade other tissues and to metastasize. But, do you know the events of invasion and metastasis? So, let's discuss the Invasion Metastasis Cascade. Firstly, there is local invasion. Then, there is intravasion of adjacent blood and lymph vessels. Then, there is transit through blood and lymphatic systems. Then, there is extravasion into parenchyma of distant tissue. After that, there is formation of micrometastases. But, do you know what is micrometastases? Well, small numbers of cancer cells that have spread from the primary tumor to other parts of the body, and are too few to be picked up in a screening or diagnostic test. Lastly, there is colonization as macroscopic tumors. Moving to seventh hallmark of cancer, which is reprogramming of cellular energy metabolism. Under aerobic conditions, normal cells process glucose to pyruvate through glycolysis. But, under anaerobic conditions, glycolysis is normally favored, and relatively little pyruvate is produced. In both, ATP is gained. However, even in well-oxygenated cancer cells, glucose metabolism is restricted largely to glycolysis. This involves an 18-fold reduction in ATP production. Moving to last hallmark of cancer, which is evasion of immune destruction. Our immune system eliminates possibly 100,000 potential cancer cells every day. But successful cancer cells have found ways to evade immune surveillance and destruction. They flourish best in immunocompromised hosts, including HIV and pharmacologically suppressed patients deficient in T and B cells. Cancer cells paralyze infiltrating immune cells by secreting tissue growth factor beta and other immunosuppressive factors. So, this was all. Thank you for learning. Join us again.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.